welcome back. So what we are telling you is, so now coming back where we left the problem. So we left the problem here with poly D lysine, and I told you that you can really check it out, that how its surface properties are changing, how its charge behavior will change. You can do a X-ray photoelectron spectroscopy, you can do an AFM, you can do a contact angle measurement analysis. Once you have done this with one substrate, the next thing comes, you have to now really take this. Say for example, I have 4 different concentration of poly D lysine, okay, PLD1, PLD2, PLD3, PLD4. Now comes the next set of experiments where the real life, so you have to put the cells on them along with the medium. Okay. Okay. Now, seeing their attachment properties, and you can do very simple things to see the attachment. So say for example, you have a cell suspension like this, and this blue is showing you in the medium where it is all the cells are suspended. Now once you plate the cells, you have a small thin layer of medium in and around the cells. You have this cover slip, you take the cell suspension, just stop it there. Whether, first of all, look at it whether it spreads or not, first catch. So say for example, here you have the substrate, and here you have in your small test tube, you have the cells what you have isolated, and I am showing the cell. Okay, so here you have the cells, and cells are suspended in a medium like this. Now once you put this suspension out there, either it will remain like this initially or may remain it for a while, or the other option is that it may spread out like this. Cells are shown in dark blue, okay, or it may spread even faster like this. These are the possibilities, and you can map it with respect to time. Does this one becomes like this, or this one remains like this, or this one's become like this? You really can see it under the microscope. So what you needed is that you needed a microscope out there which will give you an idea that how this molecule is interacting. Okay. Now based on this interaction, you can say how the surface is behaving, or does this data matches with your contact angle data which was done without the cell. Okay. How much similarity they have, or how much dissimilarity they have, how much close in there. Second thing what you can do, very easy ways, you take this dish, you wait for say 30 minutes, and then you just slightly tilt the dish. So this is on a dish, and you just change the angle to something like this. Just, of course, you haven't still filled the medium. If these cells are not attaching, you will see the cells will be rolling on the surface after half an hour. But if and this is I am talking all from my experience, if the cells had to attach by 15 to 30 minutes, they will attach, and still some of the cells will not attach. That you have to give it as a error margin will always be there. But if they have to attach, and if you tilt this dish a little bit, so what I am suggesting is, say for example, here is the dish where you are growing, and say for example you have a cover slip which is coated with your XYZ substrate, and you have these cells which are being here, and now you just tilt the dish a little bit like this. Little bit, very small, maybe this much, like just slight tilt. If you do a slight tilt, if these cells have attached on that substrate, then you won't see, you will still see some cells which will be rolling down. They will be, it's a very nice sight to see. They will be hopping down, rolling down, but still you will see cells which are more or less, looks to you as if they are almost kind of sticking to that surface. And at this stage it is very interesting. At this stage, if one can really figure out some way or other, one will be able to see that these cells have started secreting extracellular matrix on the surface. Okay. So coming back, so based on these small cues, you will be able to figure out whether the cells are properly adhering on that substrate or not. And the thumb rule is, after 30 minutes or sometime even I have gone up to 1 hour, you then fill the whole dish with the rest of the medium and put it in the CO2 incubator for growth. If it is a CO2 dependent, CO2 incubator is needed, or you can also grow it in simple air incubator, depending on what kind of medium you are using and what kind of buffering. We'll come later into that whole concept of buffering and incubator, kind of incubator what you can use. But at this point, just accept the point, you just take it and put it in the incubator in a sterile environment to grow. So based on the amount of poly-D lysine, you can predict whether the poly-D lysine is helping the cell to adhere or not helping the cells to adhere. And as much, if I assume that this is how the dose is changing, then this particular zone, what I am telling you, is where you may not see an attachment. At least from my experience with neural cell, I have seen that these are the zones, even if they attach, eventual growth is kind of very funky kind of growth. So with poly-D lysine, a very low concentration does work, and the way it works, it is believed to be these positive charges which are on the surface of poly-D lysine, these positive charges from NH2 groups which are present there interact with the surface negative charge of the cell, possibly. This is what is believed. It's a purely in the initial phases, it is purely a electrostatic interaction. And this electrostatic interaction helps the cell to settle down at a substrate, where I am telling you this part. It helps them to kind of build that first level of, so this is that electrostatic interaction which is kind of keeping them at the substrate, and followed by that it gets that time window to settle down. It's like you get a little of a time, and like there is a slope or there is something you are moving, but you just get little time to hold on to a point, and then in the meantime what you do, you tell your genetic machinery, you start producing extracellular matrix at that substrate. to get a better anchorage on that. So these pink what you are seeing are the ECM secreted by the cell. Okay. So what you see here is one can monitor this whole process in a very systematic way, which otherwise if you look at most of the cell culture work currently happening across the world are not being done. But these becomes more and more important as we talk about using cell-based biosensors. We talked about using microfluidic devices, cell-based microfluidic devices. We talk about, lab on a chip. So these are some of the emerging technologies like, lab on a chip, cell-based biosensors. All these things will require a very, very thorough surface analysis before one can really translate these technologies to the industry grade level of drug screening and other things. We'll come later into that part why I'm highlighting that part, because there are issues which are being faced by people while trying to repeat some of these results in different parts of the world, and then they realize that it is kind of, what I should say, it's kind of very incoherent results which people meet. Okay. So now we talked about the interaction which is possibly happening between polydilysine and collagen, polydilysine and the cell which are present there. And so there are two levels. So first, again just to enumerate, the first is a cell out here, substrate out here, and first is the electrostatic interaction, step 1, followed by 2, which will have where the cell upon that stabilization starts to secrete out its own extra cellular matrix or ECM. Okay. So keeping this paradigm in mind, we will move on to, and of course we talked about the interaction between, this is the kind of interactions which are possibly happening. So next in that line, now having said this, I will take a synthetic situation, sorry, a natural situation. Okay. So I've talked about these two. Now let's talk about a natural molecule. So if we talk about, if we go back to our list, so we talked about this. Now let's talk about collagen. In the case of collagen, again you are doing the same thing. Okay. You take a substrate and you have the collagen protein dissolved in a buffer. So here you are not using any more water, you may have to use a buffer. Here is a collagen protein. Okay. And here you have the buffer in which this is dissolved. And now again you follow the same thing. You create at different concentration. You have the collagen already getting there, and along with a small amount of buffer. Now what you do is, once you coat the substrate, so in terms of collagen, collagen could be used at different concentrations. You can make a, one second, you can make a thin film of collagen, you can make a thin gel of collagen, and you can make a thick gel of collagen. You have lot of possibilities what you can play around with collagen. How you play with this? The way you play is out here. So for example, you have this molecule out here. Now, if you have to play it as a thin film, then you have to have minimalistic number of molecule or minimum number of molecule, and you should be able to spread it out all over the substrate the way you see it here. Okay? So it forms a thin film out here. Now you wanted to make a gel out of it. Very simple. If you want to make a gel out of it, what you do is this. Thin gel, you are making a thin gel. So increasing the concentration. So in other words, what you are doing, you are increasing the, you are introducing a depth feature by increasing the concentration of collagen, and thereby you are bringing more collagen molecule close in. So there will be two level of interaction. One interaction you have to made happen is between the individual collagen molecules. There is one set of interaction which will be happening that will bring them close, whereas there is a second level of interaction which is the surface which is exposed, which I am showing like this. This is the surface which is very important for you because this is where the cells are going to interact. So the black, what I am drawing are the cells. So this interaction, what will be happening here is very critical. Second thing followed when you wanted to make a thick gel. Say for example, you wanted the whole matrix to be a gel matrix. So which will be something like much more complex interaction which will be coming through, something like this. Now whenever you are trying to do this kind of things, you are giving much more. So what you, if you compare these two, this picture and this picture, compare. If you compare these two pictures, what will you observe is the thickness has gone up. The thickness has gone up. Then there are two aspects which will come into play. The first aspect which will be coming into play is what will be the space between these individual molecules of collagen. So here you are no more growing the cells not on a glass substrate with thin film or a a thin film or a thin gel. You are having the whole gel and you want to grow the cells on the gel. In other words, you wanted to make a three-dimensional culture. And if you wanted to make a three-dimensional culture, then there are few other parameters which has to be taken into account. And we'll just continue in another, with few slight changes, we'll continue from here. Okay, thank you.